It's a great pleasure to be here and talk a little bit about the work that we have been doing with Seneca at South Dakota State. Fabio asked me to give a brief overview of what we have learned so far with this virus that emerged around the world in 2014, 2015. So just an outline of the talk. I'm going to be talking a little bit about the emergence of the virus, what we have learned, what we have done in terms of research at South Dakota State to understand a little bit more about this virus and the future perspectives, or what we think we should do in the future regarding this important viral pathogen. Along the years, the swine industry has experienced the emergence of several important viral pathogens, and there's a whole list here. And the first virus on the list is obviously PRRS, which emerged in around 89, early 90s in U.S. and in Europe. Another important virus is PCV2 that emerged in 1997. Then we have H1N1 pandemic flu in 2009, PEDV and porcine delta coronavirus in 2013 and 2014, and in 2015 we experienced the emergence of Senecavirus here in U.S. It had originally emerged in 2014 in Brazil, and a few months later we started observing increased number of cases here in U.S. The way that the disease actually manifested when the virus first emerged was very scary because it looked like foot and mouth disease. And those are some pictures that were sent to me by Dr. Luizinho Caron from Embrapa in Brazil. And those were some of the first outbreaks that they have seen there. And you can see this sow here with a very enlarged vesicle on the snout with a significant amount of fluid on those vesicles. And it's very difficult to distinguish from foot and mouth disease. Here are other pictures showing the first, some of the first outbreaks here in U.S., also showing those vesicular lesions on the coronary bands and on the snout of affected animals. So it was really scary because of the similarity with foot and mouth disease. The clinical signs that these animals presented included lethargy and lameness, obviously because of the location of those lesions in the feet. Morbidity was variable. There were herds that reported 5% morbidity, 5% of the animals affected with the lesions, to up to 75% in some cases. Another clinical situation that was reported and associated with Seneca was neonatal mortality. This manifested by lethargy and anorexia on piglets. Animals would also present diarrhea. 50% of the piglets would present diarrhea. Morbidity also varying between 10 to 75%. The difference here is that a high rate of mortality was observed between 15 and 50% mortality in those piglets. And those piglets that actually died, they presented high viral loads in different tissues. The duration of this mortality and diarrhea outbreaks usually was between 4 to 10 days, and then the animals, the other leaders would be normal. One of the features that was observed, and this is all field observations, was that you would see leaders affected with mortality. You would lose the entire litter on a sow, and the litter that was on the next crate on the side would be normal. The relationship of Seneca with neonatal mortality and this form of presentation has not been experimentally demonstrated. So far, we don't know if this was really caused by the virus, but it was a field observation that you would see the sows presenting vesicular lesions and mortality on the piglets. But nobody was able to reproduce, or there's no published literature demonstrating the implication of Seneca with this form of presentation. So this is when the disease was discovered in Brazil in 2014. So it was September of 2014 was the first description or diagnosis of the disease. And it quickly spread to, affecting up to 70 to 80% of the swine industry in Brazil. And a few months later, in July of 2015, we detected the first outbreaks here in the U.S. And I think the first outbreak was actually diagnosed in South Dakota. This is data from the Swine Health Monitoring Project that encompasses data from July of 2015 until August of 18. And you can see that we had some peaks here in the beginning, and then in the following year, usually in the summer months, and right now we are kind of in a decline in the number of outbreaks being reported here in the U.S. But one of the interesting things is that before 2015, this virus was already circulating here. There's evidence that the virus has been circulating in swine in the U.S. since 1988. And there were only 1 or 2 reports of the virus every year until 2015, when we observed this significant increase in the number of cases. And up to date, we have about 300 cases of Seneca that have been confirmed and reported to the Swine Health Monitoring Project. There's probably many more cases that have not been reported, and we know of several of those cases that are not part of those official statistics. So what did we know about the virus before or when it emerged in 2015? So we knew that it was a picornavirus that had a genome of approximately 7.2 kilobase in size. And the virus had been associated with sporadic cases of vesicular disease in swine, 1 or 2 cases a year, as I just mentioned on the previous slide. This virus was actually first isolated in 2002 as a cell culture contaminant. This was a company that was working with human cancer cell lines, and they actually detected this cytopathic virus in their cell culture. They sequenced the virus, and they identified this new picornavirus that they classified as a new genus in the family Picornaviridae, the Senecavirus genus. After that first description and the complete genome sequence of that virus, some people from UK, Pirbright, in collaboration with NVSL and NADC, went back and sequenced some archived swine samples that they had collected since early 1980s, because there was a picornavirus associated with those samples, but they didn't know what the virus was. So they went back, sequenced it, and detected Senecavirus in those samples, showing that the virus was circulating here since late 80s. And that's the first description, or the first isolate that has been detected in U.S. since dates back to 1988. Another thing that we knew is that experimental inoculations with the historical strains, with those older strains that isolated before 2002, they did not induce vesicular disease in pigs. So people had tried to reproduce the vesicular disease, and they were not successful on that. So the causal relationship of Seneca with vesicular disease was still unknown, although the virus was associated with several of those outbreaks. So this is just a comparison between FMDV here. This animal is infected with FMDV, presenting that vesicular lesion on the snout and the coronary bands. And then here you can see an animal infected with Senecavirus. In the field, it's impossible to distinguish those two. That's why this disease is important, and I think we should continue to do research to understand more about this virus. So what were the gaps in knowledge then when the virus emerged in US? We didn't know much about the genetic diversity of the virus. Why did we actually see this sudden increase in the number of outbreaks? Was it genetically related? Were these new strains that were circulating different from the older strains? We didn't know that. We didn't know if the virus was actually the sole cause of vesicular disease. The Cox postulates had not been fulfilled before. We didn't know anything about the pathogenesis of the virus, neither about immunity, cross-immunity between different strains. Do we have maybe a new serotype that emerged, and that's why we're seeing these increased outbreaks? We didn't know anything about the prevalence of the disease and nothing about prevention and control. How do we deal with this? So those were pretty significant gaps that were not known, that we actually needed to fill through research. One of the first experiments that we did, and that's how I got involved with Seneca, was through a project funded by SCHIC to determine the genetic diversity of Seneca isolates. This is a pretty large phylogenetic tree. This work is going to probably be submitted for publication in the next few months. It involved collaboration of several different universities and places. So the idea was to sequence contemporary isolates, determine the complete genome sequence of contemporary isolates circulating in the US and in Brazil. And here we have the phylogenetic tree of 113 complete genomes. About half of those were determined by our group and Dr. Marthaler and some people from NVSL. And the other half are sequences that are available on GeneBank. As you can see, there's a clear separation between the historical strains, those isolated between 2002 and 88 down here, from the new strains that emerged in 2015 in US. Another interesting thing to note on this phylogenetic tree is that it seems like the viruses that are circulating in different countries, they form distinct phylogenetic clusters, which means that they're sort of evolving independently from each other. Another interesting thing is the sequences that are available from China. They cluster, the majority of them cluster here in this separate branch in the tree. But there's also several Chinese strains that are spread out throughout the phylogenetic tree. We don't know exactly what that means. It could mean that the virus is actually going back and forth from one country to the other, but it could be just incidental evolution of the virus independently in different places of the world. This is something that it's pretty striking and is interesting. It happened only with the Chinese strains, and here there's a strain that also is an outlier of this tree, which is a strain that was detected in Colombia in 2017. So that was what got us started on Seneca. And then the second major break in research on this was done by Dr. Kelly Lager at NADC, where they actually were able to demonstrate that Seneca was indeed the virus that was causing disease. So they took an isolate, they inoculated pigs, and they showed that those animals would develop vesicular lesions just like the ones that were being observed in those field outbreaks. So we did a follow-up study. I think we were kind of working simultaneously or in parallel with those two viruses. And what we did is we also took an isolate, one of the contemporary SVA isolates, and we did inoculation in pigs to assess a few more details about the pathogenesis of the virus. How is the virus actually causing vesicular disease? So what we learned on that study is that usually animals would start presenting signs of vesicle formation around days 3 to 4 post-inoculation. Those vesicles will progress to fluid-filled vesicles around days 5, and 6. Then those lesions will eventually rupture. We will see development of scar around day 9. By day scabs, by day 11, we would only see a scab on the area that was affected. And usually by day 14, 15 post-infection, those lesions will be completely resolved. So that was the clinical picture, and this is pretty consistent. There's usually 1 or 2 days difference between each of the animals in the experiment. We also did a lot of work in terms of assessing viremia, virus shedding. We found out that the virus induces only a pretty short viremia between days 3 and day 10. Post-inoculation, after that, we didn't detect any viral RNA by PCR in serum. Virus shedding was performed or assessed in nasal swabs, oral swabs, and rectal swabs. We detected shedding up until day 28 in all those 3 samples. After that, no shedding was detected anymore. Then we did virus isolation, and also this is a PCR data. We did virus isolation to assess infectious virus, and what we found is infectious virus shedding up until day 7 on nasal swabs, up until day 14 in rectal swabs, and 21 on oral swabs collected from those animals. We also did test several tissues for the presence of viral RNA. And we so we euthanized 1 animal per day between days 3 and day 7, and then tested all these tissues for the presence of viral RNA. What was striking to us is that all the animals euthanized between days 3 and day 7 presented pretty significant or high viral loads in the tonsil. And then when we ended the experiment on day 38, we also assessed presence of the virus in those tissues. And again, the tonsil called our attention because there was a significant amount of virus there. And all the animals, so here we had only 1 or 2 animals positive in those tissues. And here in the tonsil, all animals were still positive at the end of the experiment on day 38. This is like 3 or 4 weeks after complete resolution of the disease. This is something that we don't know exactly what it means, but it could be persistence. And that's something that we are currently working on and investigating. Why would these viruses or these animals still have this amount of viral RNA in the tonsil so long after the resolution of the disease? So that's an aspect of the virus that we don't completely understand yet. So we also did studies on immunity, and what we saw is that there is pretty significant, this is a virus neutralization assay. We detect pretty robust and very early neutralizing antibody responses. So by day 5, all the animals are inoculated. They already have these pretty high titers of neutralizing antibodies. And peak neutralizing antibodies usually around day 7, and those will be maintained for several days after inoculation. We did a quick correlation between levels of neutralizing antibodies and then levels of viremia, and there is a strong negative correlation between increased levels of antibodies and decreased levels of viremia, suggesting that those antibodies are, sort of controlling that viremia and helping the animal to fight the disease. We also did some IgM and IgG isotyping to characterize a little bit more that response. And we saw that IgM antibodies are the ones that are appearing earlier. By day 5, we detect IgM antibodies, whereas IgG antibodies show up a little later. There's, again, a strong correlation between IgM, and the neutralizing antibody titers that we detected on day 5. We did some work on T-cell responses as well. And, again, we detected pretty robust and early T-cell responses for any viral infection. By day 7, basically all the CD3, CD4, CD8 T-cell responses that we evaluated were enhanced in the animals that were infected. And even by day 35, which was the last day of the experiment, we still detected that. So it's pretty interesting that the animals develop these T-cell responses and we still detect a viral RNA in the tonsil. So somehow the virus is probably manipulating and evading those responses to be able to remain in the tissues of affected animals. This is one of the most recent studies that we did, which was a comparison between a historical and a contemporary strain. So we took that isolate from 2002 that was obtained from cell culture, as a cell culture contaminant. Most likely, the suspicion is that that virus probably came from porcine trypsin that is used in cell culture. So we took that virus and we compared it in finishing pigs to one of our contemporary isolates, the strain SD1526. And what we saw is that animals inoculated with the historical virus did not present any evidence or any clinical signs or lesions associated with Senecavirus. They remained normal, clinically normal throughout the experiment. Here's just a graphic representation of that, where we have clinical scars. We score the lesions that we observed. In blue is the contemporary strain. We can see that most animals developed vesicular lesions, and all the animals inoculated with that SVV001 strain remained clinically normal throughout the experiment. We also did compare viremia levels, shedding in oral, nasal swab, and fecal swabs, and you can see that there is significant differences in viremia and also in viral shedding in nasal swabs and oral swabs between the two viruses. These results actually suggest that this virus, this particular strain, is a low virulent strain for pigs. We also did cross-neutralization assays to see if there is any differences in the neutralizing responses against these two viruses, and cross T-cell responses as well. When we took serum from, when we actually tested serum from animals immunized with SVV0 or challenged with SVV001 and challenged with SVA SD1526, we tested them against the SVV001 strain. We saw pretty similar neutralizing antibody titers. So these results actually suggest that at least between these two strains, we're talking about the same serotype. These two genetically diverse and historically separated strains, we have one serotype. There's pretty significant cross-neutralization between the two. We also did the same with T-cell responses and the levels of responses when we stimulated PBMCs collected from both groups with the heterologous strain or the homologous strain was pretty similar, showing conserved T and B cell epitopes between these two strains. So in summary, what we learned with all these studies is that the virus actually induces a pretty short clinical disease, self-limiting disease that will be resolved between 12 and 14 days post-inoculation. There's a short-term viremia between days 1 and 10 post-inoculation. We see pretty significant humoral immune responses or antibody responses early on, and most of those are neutralizing antibody responses around days 5 to 7. Animals also develop significant and pronounced T-cell responses. Virus shedding is detected up until days 21 to 28, with infectious virus being shedded for 7 to 21 days, depending on the secretion that you assess. It seems like that there's a pretty obvious interplay between the immune responses here and resolution of the disease. As the immune responses are detected, we see a decline in viremia, decline in virus shedding, and clinical resolution of the disease. We still don't know what is the exact function of each of these components of the immune system to that, but there's pretty nice correlation between the immune responses and resolution of the disease. So what do we still need to learn about Seneca? One of the first things is prevalence. I mean, we don't know what is the prevalence of this virus in US, and we don't know about the prevalence in other major swine-producing countries as well. So that's one very important question that is still open. What are the reservoirs? Are pigs the only reservoir, or do we have something else that actually carries the virus and transmits the virus to pigs? We don't know that. In one of our first studies, we actually detected the virus in mice and in flies, that were collected in an affected farm. We were able to isolate virus from mice, but we don't know if mice are actually a natural reservoir or not. It could be just virus passing through the GI tract. We actually isolated the virus from the intestine of these mice. But that's something that we don't know. They could function as reservoirs. Wild boars, I know of some people that actually detected the virus through metagenomic sequence in samples that they collected from wild boars. Could they play a role? Nobody knows about that. What's the role of feed on transmission? So that's another big question. I mean, we did some work with Dr. Scott D. We showed that the virus actually survives in feed, several feed ingredients, for up to 37 days, with very minimal reductions in titers after we spike the feed at the end of those 37-day incubation periods. So that's another important aspect that probably needs to be addressed and looked on. We don't know anything about molecular pathogenesis. We don't know anything about persistence. Is that true persistence? Are these animals that carry the virus in the tonsil, are they able to shed and transmit to other animals? We don't know that. Role of stress on disease outbreaks. There are some associations between transportation to slaughterhouses and outbreaks of Seneca. There's also associations between parturition and outbreaks of Seneca. Those were the initial descriptions. We don't know if these are actually playing a role on the disease or not. So that's something that probably somebody should look at. We don't know what the protective or the effector mechanisms of protection are. We know that there is pretty robust neutralizing antibodies and T-cell responses, but what is the exact function of those in protection is still unknown. And as far as I know, there are no vaccines available, although I heard that there was a talk yesterday about a potential inactivated vaccine that has been or is under development by one of the big vaccine companies. So those are still things that probably need to be addressed. And the main reason why they need to be addressed is because we really cannot tell the difference between Seneca and FMD. And we probably don't want to live with that doubt when we see an animal with a vesicular lesion. And I think this will be even more important should FMD be introduced in this country. Is it Seneca or is it FMD? So with that, just a little bit more data. I talked a little bit about surveillance, and this is a preliminary, very preliminary study that one of my undergrads just completed a couple of weeks ago. So she took about 1,000 samples, serum samples from 10 states in US and also 50 samples from Manitoba and tested them by neutralizing assays. And what we saw is that we have an overall prevalence of about 7.6. So 7.6% of the samples were positive. It's not very high, but it's an indication that the virus is out there, is circulating, probably quietly, and eventually will cause outbreaks here and there. With that, I would like to thank you all for your attention. Thank you again for Dr. Panucci for the invitation and for the great collaboration that we have established on this area of research.